Well, hello, and it is quite bizarre at this end looking at a camera, so I'll have to imagine now an audience of thousands in front of me so I can actually give you a proper presentation. I'd like to tell you something about the company and something about the science, not too deep, just enough to give you an overview of what we're doing. And then, because this is an interim statement discussion, have a few slides at the end which will allow me to expand a little on the interim statement we put out. The finances, as you probably have seen and have found from reading, are a little unusual because of the nature of a reverse acquisition. But if I start off by taking you through the general strategy of the company, and what we are set up to do is to discover and develop new molecules which can address metastatic breast cancer. Now that's the primary indication, but the technology that we're developing this from has applicability in a number of other cancer areas. So we want to first have a proof of concept in breast cancer, and then, funds willing, expand that into other areas. We have also, since the time of the IPO in April, we have in-licensed some complementary diagnostic technology, and I'll tell you a little about that. And overall, we do operate a pretty lean and virtual business model. We outsource our research to Cardiff, where the experts are, and we don't intend to have too much in the way of central costs. And the general idea of that is to have the funds that we have from our investors applied to the R&D and not to some unnecessary infrastructure. So, if you have visibility of the slides, I'll try and indicate where I'm changing slides and different topics. But I'll overall talk about the people involved, and I'll then go on to tell you a little about the Cardiff research, the deep into the science, and we'll then, I think, give you a good idea of the company. So if I start with the people. Now, if I take the management first, the few of us, there's myself and Gabriele, primarily at the moment. Gabriele, a lot of experience in founding and funding biotech companies, raised significant sums of money for his companies, listed 5 on NASDAQ. We have Tiziana in the UK, listed on AIM, and a few other private companies, all in the early stages of development. So with that background and history and the successful exits, particularly in 2012 when Gabriele managed to sell to Bristol Myers Squibb, one of his companies, Inhibitex, for multiple $billion, that gives us a good track record of getting the money to be able to do this research. For myself, I've had an operational focus for the last few years. I started off as a scientist with a PhD in genetics, molecular biology, but found I didn't have quite the craft skills that you need to be good in the laboratory, so switched over to finance and had a business training in Unilever, Kraft Foods, and in consulting until about 15 years ago. I took my first job as a finance director back in where my heart is, biotechnology, and looking at mixing the business skills I have with the science that I so love. So with that, I've, in the last few years, worked for Syntaxin, which was a venture capital-backed biotech, and we successfully sold that to Ipsen Pharmaceuticals last year for upwards of 150 million euros. And with that operational knowledge and the science, which I wouldn't say is expert science, being so long out of the laboratory, I have the ability to advise the scientists on how to take their discovery and their development forward. Should we get to a point where we need to appoint other more expert development scientists, for example, we'll do so, but again on the advisory and virtual model. The other persons on the board, we have Andrew Gutman. We inherited from the former ADI, Alexander David Investments Board, and that gives us good continuity for the listed company. And with his financial expertise allows us to have that insight into the public markets and investor base. And then we have Ricardo Dalla Favra, and he comes with a wealth of experience of clinical oncology from Columbia University, and gives us that insight into the science side on the board, in addition to the scientific advisory board. The triumvirate in the middle of the slide are the scientists who discovered and developed this technology. And what I liked about this when I saw the guys was that we had three complementary skill sets. A skill set, firstly, from Andrea, who looks at the computer-aided design of molecules, of computer-aided drug design. We had Andrew Westwell, a medicinal chemist, looking at how to make those molecules. And then we have Richard Clarkson, whose skills are in the biology of cancer, particularly stem cells, and in particular breast cancer. And he has the models which allow those designed molecules to be tested. So we've got a very good combined integrated core of researchers who work very well together and who are busy taking the investors' money and using that well to design new molecules against the target. On the right of the slide, we have the Scientific Advisory Board. And this is a group of very eminent scientists, which we're very pleased to have attracted to Tiziana over the last 6 to 12 months. We have Napoleone Ferrara, who was the inventor of Avastin, one of the biggest selling cancer drugs out there, at $3 billion of sales per year. And he's seen that drug through from its early discovery to the development and the marketing. So he's able to provide those insights to the scientists and to the Tiziana team in general. We have Roberto Pellicciari, who is a medicinal chemist of renown and founder and still a chief scientist for Intercept, a $5 billion listed NASDAQ company. And there we have the advice on medicinal chemistry that the Cardiff team can take and learn from. And then we have Professor De Fiori, who has, as well as providing us with the technology around the diagnostic that I'll mention later, is able to provide, again, good oncology expertise and drug discovery and development expertise. So you can see that with a very virtual team, we're able to attract a great deal of skill into the overall company. So, if I tell you a little about the research, well, biotech research is fundamentally predicated on good intellectual property. And the foundational IP for intellectual property IP for this research is around the BCL3 gene, as it's known. And this gene codes for a particular protein. And it's this protein and its action that the team first elucidated in Cardiff and were able to get patent protection upon. We've applied for the GB application, went in July 13. That converted to a European patent and is due to publish in January 15. And we've also put in a provisional patent application in the US. And, of course, always look for other patenting opportunities. And the reason we're able to put that in is whilst the BCL3 gene had been known about, its action or involvement in metastasis, that's the movement of the cancer cells, hadn't been spotted. So we're able to get a protection on an area, and it's a double-edged sword. In a new area, you have great opportunities, but there's also a lot of biology that isn't known and is still being developed by various laboratories around the world. So the team in Cardiff is keeping on top of that, keeping on top of the different angles of BCL3 and metastatic cancer research to bolster the company's knowledge. And if I talk generally on the next slide about cancer and the particular treatments that all of the companies around the world are trying to put in place. In previous decades, the emphasis has been really about tissue, about a cancer treatment for a particular cancer, be it breast, be it lung cancer, be it brain cancer, whatever. It's been defined by the tissue. over the last 10, 15 years or so, it's been defined far more on what's been termed epigenetics, defined by the pathway of the cancer, how that cancer develops. And this is where the Tiziana research is focused. Not so much on the pure oncogenesis, the formation of the cancer in the first place, but on what makes the cancer go from being a solid tumor to one that spreads around the body. And the fundamental premise that we have is that if it's starting to move around the body, then you're chasing a moving target, and whatever treatment you're trying to do, you'll have a lower chance of increasing survival just because you're chasing that cancer. If we can find a mechanism that impacts on cell motility, cell movement, and therefore keeps that cancer caged in one place, then whatever treatments there are have a better chance of success. And then, in the end, what we're all about is increasing life expectancy and using the statistical 5-year survival rates, increasing those survival rates so that we improve patients' life. And that's the fundamental business of biotechnology, to improve patient livelihoods. So, cell motility being the area doesn't necessarily just apply to breast cancer. We chose breast cancer as our proof of concept because of the expertise of the Cardiff team, and it's a good and unaddressed market. But we can always take that science, once we've proven it in breast cancer, and expand into other cancers that have the same metastatic issue. And that gives increased value to the company. But as you always find with biotechs, we are constrained by the amount of funds. We'll focus the funds for the moment on breast. When more funds are available, when the proof of concept is there to allow those funds in, then we can expand. And some of the broad statistics on breast cancer. I mean, it's known that over the last 30, 40 years, diagnosis and detection have improved tremendously through not just pure knowledge and awareness and the mammography, scanning. All of these things are great for the early stage detection. But inevitably, cancers progress, others are not spotted, and there's a typical staging in cancer where you move up the stages from 1 to 4, and in each stage your 5-year life expectancy reduces. And whilst there's been improvements in the survival in the first 3 stages through the improved drugs we have nowadays, stage 4, when we have metastasis and the movement of the cancers around the body, is still a very poorly addressed need, and one that we want to focus on. So if we can slow or stop the cancers moving into that stage 4, we will by default have improved survival. Now if I give you a little bit of science, and when I'm giving this sort of presentation in front of an audience, at least I can have people chip in and say, stop, you're going a little bit too fast, or it's too complicated. Hopefully this will be at a level that's not patronizing, but also one that gives you an explanation. And the first slide is really about the pathway. And all the takeaway from this slide is that the particular gene that we have, BCL3, and the protein that we're trying to inhibit is part of the pathway. And if we can inhibit that gene and inhibit its protein product, then we'll be inhibiting that particular pathway. And on the right-hand side of the slide that you might see, when BCL3 forms a complex, it allows movement, and when it's not in complex, the movement doesn't happen. Therefore, by inhibiting the complex formation, the idea is to inhibit the movement. And to give you a little anecdote, I liked, when I was growing up, I watched a program on television called Tomorrow's World, and it had very visual explanations of science. And what I find when I look at science as a CFO, if I can see something that is simple and self-explanatory perhaps, then that gives me greater confidence in the science base in the company. And when I saw this for the first time at Tiziana, it was very, very straightforward. And on the slide that you should have in front of you now are two molecular structures, both of the BCL3 protein. And this is where the team at Cardiff really showed their skills and showed the serendipity of science. They saw the molecule and they thought, well, what would happen if they designed molecules that would fit within the pocket that they spotted within the molecule? So they designed some molecules in the computer-generated design, and put those molecules into the pocket. And it appeared to inhibit the complex formation. So they took some of those molecules and tried to make them chemically. And in making them chemically, they were then able to test, using various experiments, in vitro, in the what they say in the test tube or in the Petri dish, and to see if that had the same inhibitory effect. And indeed it did. And so they were then able to take those same molecules and try them in vivo, in animal testing, in small mice. And again, to show that the cancer-inhibiting movement effect was seen. So it all came from an understanding of the structure of the molecule and a structure of the chemicals that could inhibit that molecule. Quite straightforward science, but of course very complex to actually put into action. So one of the tests you do is, if you don't just have an idea of what might happen, you say, well, what would happen if we were in an animal that didn't have BCL3? And there are strains of mice that have been bred to not have BCL3 gene. And if they don't have the BCL3 gene, they're healthy, but they can't produce that BCL3 protein product. And when tested by injecting cancer cells into those mice, they saw that there was much reduced metastasis by not having BCL3 gene in those mice. And that was the first control experiment they could see. If you take to the next slide, you have genetic suppression, another way. So this time you have mice which have the gene, but you genetically suppress the expression of that gene. So again, you don't get the protein. And once again, in the experiment, they showed much reduced metastasis. Another tick in the box for the science. And then finally, a panel of mice that we can visualize using certain imaging techniques to show the metastasis. They injected the tail vein with cancer cells, showed how those moved around and formed tumors in control experiments. But then in mice that had had at the same time an injection of the molecule, the anti-BCL3 molecule, there again was a much reduced metastasis. In the lower panel, only one mouse had localized tumors around the tail. The other mice in the top panel showed multiple tumors in different organs around the body. So with these in silico, that's in the computer, in vitro, in the Petri dish or in the test tube, and in vivo, in the mice experiments, we were seeing proof of the action of BCL3 on cell movement of cancer cells around the body. And it's from there that the team around March, April time, coincident with the reverse acquisition, were starting to increase the research onto which molecules they might choose to take forward as a clinical candidate. So for the last 6 months, they've been designing new molecules with slightly enhanced binding or into that pocket, trying to find the molecule which has an optimum set of properties that they can then take forward to further tests, and then ultimately in the next 1 to 2 years take forward into the clinic. And for that, of course, you've got to do early toxicology, preclinical toxicology, but in the first instance you've got to make more of the molecule. So all of that drug design, drug development is going on at the moment, and looking to choose that first clinical candidate. A small timeline, very rough at this stage. There's a lot of experimental design in the background behind this, but you'll understand that research is not a purely linear process. So as the experimental results come out and other experiments are designed, you have to adapt around that main route. But we're confident at the moment these particular timelines will be fulfilled. And so what we're looking at is choosing that clinical candidate, possibly by Q1 next year, Q1 15. Then we will be designing further experiments and engaging with manufacturers to make the molecule, because that has to be made in high quality for clinical trials. And then engaging with other contractors who are experts in the sort of preclinical toxicology that we'll need to do to have all of that proof and dossier for the regulators ready for that first clinical trial, targeted for the end of 2016, beginning of 2017. Excuse me while I just refresh my quote. So, whilst the BCL3 project is our main thrust, we're always looking for other opportunities to increase shareholder value, to complement that research. And one particular area which complements very nicely is this technology that we in-licensed for, which is termed Top 20. And it's a technology that identifies in the blood particular signals of the top 20 genes in cancer, and by looking at the profiles of those is able to stratify patients into different categories who may or may not develop more aggressively or who may or may not respond to certain treatments. And the idea for this is, as many areas of research development in the life sciences at the moment, is to try and make better use of that health dollar and spend it more wisely in patients that will respond, or treat patients more urgently if they need to be, if they're of a risk group that needs that more urgent treatment. And the other aspect of this is that it's based around stem cell technology, and the Cardiff team have expertise around that, being based out of the European Stem Cell Institute in Cardiff. So it's a nice intersection of the different sciences and complements a therapeutic approach with a diagnostic approach as well. And whilst we will always say that this is the current strategy, we will keep our eyes open for other complementary technologies or opportunities to invest shareholder funds wisely for a better scientific result and a better share result. So if I summarise in the next slide, we have novel technology with the BCL3 inhibitors. We have an unmet medical need in breast cancer, metastasis in breast cancer, but we can expand that to other cancers. We've got good IP protection to date around this particular area, and we have the diagnostic to complement. So all of that led by, without being too immodest, an experienced management team which knows about drug development from the scientific angle, knows about company development and operational issues. We should place ourselves well to take this company forward. If I pause now and move to the finances that you'll have seen in the interim result. I don't want to trouble you with too much, because I think we spent quite a lot of time putting that interim result together, and it may not have seemed it, but reverse acquisition and the accounting for a reverse acquisition is quite prescribed and quite complex, and I have to be honest, I don't think necessarily for the best shareholder information, because it doesn't allow you to give comparables. But the more I looked into it, the more it made sense, because by Tiziana Pharma being acquired by Alexander David Investments, it was a true reverse acquisition, because in effect Tiziana Pharma Limited controlled the new group. So for that reason there shouldn't be comparables, because the history of ADI is somewhat irrelevant for the development of Tiziana going forward. And to compare the financials would give a misleading impression. So I was won over by the accounting treatment in that respect. but I wanted to try and demystify a few of the items. And to that end, I'm just going to show two slides on the income statement and the financial position. And that's for the old timers amongst us, the profit and loss and the balance sheet. You can pick up all of the key items from the interim statement itself, the loss for the period up to 30th of June, and the loss per share in the calculation, and of course the cash position. Just to show on the cash position, we had 3.55 million at the end of June, and at the end of September we were looking at just about 3 million. And this will give you a better idea of the indicative burn, and even that $500,000 burn for Q3, a lot of that was taken up by payments for the remaining invoices around the acquisition itself. So the first thing to show you then would be the income statement. So I move to that. And yes, very plain, 2 columns of zeros with no comparables. And if we concentrate therefore on the 30th of June 14 column, in research and development, and that is the main research and development of Tiziana Pharma, from January as a standalone company, and then from the group from April 24. So that gives you an indication of the sort of low-level R&D we're talking about. We're outsourced, so we're only having outsourced programs to fund, but this is at the early stage of research. And as our animal models are enhanced, as what they call assays, the tests are needed, we will start seeing an increased R&D spend in the next 6 months, and then through into 2015. And then once we have contracted with manufacturers to make increased quantities for both preclinical toxicology and the final clinical trial, then of course, then you'll see higher R&D spend. Operating expenses looks like a large figure, but the majority of that was the legal and professional services expenses around the reverse acquisition. Some of those were from Tiziana Pharma, and then some from the group, again from the 24th of April. So it was a combination in this period of group 24th of April onwards and Tiziana Pharma alone earlier than that. And then the cost of listing. This is the one that we, in our accounting discussions before the interim, had a lot of argument about where to put it and where to place it. But in the end, it's a non-cash figure. It's a calculated figure that comes out of the particular accounting treatment that one has to put in for a reverse acquisition. And so when one's looking at either cash burn or indicative P&Ls, this is a one-off figure. It is only a non-cash figure. And then in financial expenses, we have the stamp duty from the actual reverse acquisition and some other expenses around the acquisition. So again, it's not fully indicative. I think I'll be more happy talking about the figures when it comes to the next statement for the year-end figures, because they'll be more indicative of the ongoing company when we take the 6 months from July to December onwards. So if I then look at the financial statements, the financial position or balance sheet, there we have a mix of items which will be familiar, but also items, particularly in shareholders' equity, which are again affected quite dramatically by this reverse acquisition accounting. I think the assets area is relatively self-explanatory with the normal titles, but when we look at shareholders' equity, this is where we have the wonders of the reverse acquisition coming into play. It treats Tiziana Pharma as the acquirer, and therefore the continuing entity, and that's why, as I said, we have the 0 columns. But because the share equity, nominal share and share premium are fixed figures and came from Alexander David Investments, those figures remain in the balance sheet. So you see those two as full figures of 9 and 16 million respectively, and sitting there as they would have done in the previous company incarnation. But because we're talking about Tiziana Pharma being the chosen acquirer, we have to find a way of reducing those down to a lower level, reflective of the company as it should have been. And therefore we have various accounting adjustments, non-cash again, which take it down so that what you end up with is total shareholders' equity reflective of Tiziana Pharma. So we have various adjustments and a big adjustment called other reserve, imaginatively, to bring that number down. And we have the opportunity for questions. So if there's any specific questions, I was steeped in this reverse acquisition accounting, so sadly I hope I'm able to answer those questions for you. I hope that's been relatively self-explanatory, and look forward to any questions you might have.
Thank you, Phil, and we do have a few. Since we've just covered financials, we'll stay on that theme. Can you give us an idea of the current monthly cash burn is, and given your current cash balances, where that takes you through to?
Okay, I'll start with a couple of caveats. Caveats being always based on strategy as is, and as I say, we're always looking for other opportunities. But on the current project BCL3 with the diagnostic as well, the current cash figure should take us through to early 2016. When we look at monthly cash burn, it's relatively low, I'd say. It always varies. So on average, we're talking between 50 and 100,000 a month. And I think this will be on a slightly rising trend as we go towards December, and then through next year, again on a rising trend as we invest more in the R&D, possibly take in a part-time development scientist to oversee that outsourcing. But through next year and into early 2016 should be where our money will last us.
Thanks. There was quite a lot of interest back when Dr. Ferreira joined the scientific advisory team. Can you give us some idea of what it was that attracted him to Tiziana in the first place?
I think a combination of things. It's always good to see a new area of science, a new approach, not necessarily just following every other cancer, well not every other, but a lot of cancer treatments are always cancer kill, that's the focus. This is quite unusual in that it's cancer caging or cancer metastasis inhibiting, and that's an unusual approach. So that plus the knowledge of who else was involved, the other members of the scientific advisory board, the skills of the Cardiff team and the European stem cell unit that they work from. All of those things, it's like a snowball effect of reputation and good science knowledge.
Thanks. I'm sure one you've heard, it's always tricky to answer, but what are the potential exit routes for investors, and do you expect material interest before or after the product approaches clinical trials?
Yes, it is a common question. I look at this as three possibles. Should the science develop well and investors continue to be interested and we attract more investors, then there's always the, I think, theoretical possibility of taking this company forward as an enlarged company addressing lots of different areas of science. That hasn't been the pattern for many companies to date, so I say it's theoretical. The two more common routes at the moment are licensing route, which would see a company that could better, more quickly and more comprehensively develop the science by taking one or more of the projects once the breast cancer one has proven, to take it forward in their areas. They may have an expertise in lung cancer and wish to exploit the science in lung cancer, so you would license out to them. So the licensing route would bring in licensing income and more opportunities to reinvest elsewhere, or simply take that income approach. And then there's the obvious trade sale exit, which can come at any time. It depends really on the therapeutic interest of the companies that are looking at you, and also, of course, the particular success of the science improving itself, or a competition from others. So we'll look at all of those routes, and it could come earlier than the clinical trials. It's not unknown. We've had interest from small to medium-sized European companies already, just dipping their toes in the water, asking about our science. So the fact that we've got on their radar already is good news. But that's a business development task that we'll take forward over the next 12, 18 months.
Thanks. And finally, why did you choose to reverse onto AIM as opposed to a listing on a European exchange, or indeed even US?
Good question again. Gabriele has founded and formed all of these companies that I started the presentation explaining on NASDAQ. And he just felt that he'd like to relocate himself to Europe, particularly London, and saw an opportunity that the London market, fantastic market for the world, not just for Europe, and seemed to be underrepresented of good biotech companies that he'd seen in the States, and an opportunity therefore for us to take it forward and prove a point. And I think you asked about why reverse, and reverse, well, let's be honest, this sort of story is a risky story. All biotechnology is, and to get a company onto the market, to have the opportunities of raising capital and getting to a wider investor base, but without having to go for a full roadshow, was attractive, because Gabriele has enough people interested in the companies that he's invested in, who follow his track record and wish to invest, that we were able to raise the relatively modest 2 million placing and the convertibles we've done without having to do the full-blown roadshow, and therefore a reverse acquisition, as well as being slightly lower cost, I believe, it was an attractive route.